Hey guys, this is Karan here. I live in Saudi Arabia studying 10th grade in Yambu International School. Today our topic is genetic variation within population. Okay, now to start off with this section, to get the idea of this whole title, let's go ahead and consider an example. Now you may think that by seeing one penguin, you have seen all of them. Well, let me correct you. Think penguin as human being. Ask yourself the same question, that is, can you think that seeing one person you can generalize all of them? Well, no, right? So what is the nature? There is a question I have for you today. What is the nature of genetic variation in population, and how is that variation measured by biologists? So we have to answer these two questions within the lesson, genetic variation within the population. Okay, now let's go ahead and continue with an example of penguin. Now you may have known that there might be fat penguin. Okay, in nature, there is fat penguin with, let's say, rounded body. Okay, then this might be one type. The second type would be slim penguin, and they might be tall, they might be short as well, they might be tall. Okay, and this over here is nothing more than types of phenotype. Okay, this over here is known as phenotype. Okay, now we cannot control the phenotype. The reason is it's because of genetic variation. Now what is it? What is genetic variation? Okay, so what is genetic variation? What is it? Now, well, genetic variation is stored in a population genes pool, the combination of alleles of all the individual in a population. Okay, now it turns out that each allele exists at a certain rate or frequency, as you might call it. Okay, now there is something called alleles frequency, which we just talked right now about, which types of alleles exist, which we said it exists at certain type of rate or certain genetic certain alleles frequency or certain alleles rate. Okay, rate and frequency, you can both name it together. Okay, so let's go ahead and we can see alleles frequency. Whoops. UENCY. Okay, now it's basically nothing but a measure of how basically a measure of how common a certain alleles is in the population. Okay, and there's something related to this which I'm going to reveal right now. Okay, we'll talk about it. You can basically also calculate the alleles frequency. Now, the diagram to calculate the alleles frequency is what I'm going to show you right now. So the diagram basically looks, the diagram for alleles frequency, to calculate the alleles frequency, is nothing different than a capital sigma sign. So how does that look? Well, it's a different kind of sigma sign. Well, basically the sigma sign basically looks like this, but the diagram is not structured like this, but it's not exactly like this. Okay, so the diagram is basically something like this. Okay, now let me go ahead and label them. The first at the top, we have gene pool. Okay, then over here you have genetic variation. And on the top of here, it's basically the same thing, but over here what I'm going to include is the sources. Okay, so this is how the diagram to calculate the Alice frequency or rate looks like. Now there is a procedure on how to calculate it. Okay, this is just a diagram that I gave you, but I also have to tell you on how to calculate the genetic variation. So the step 1, the first step, and what I'm going to do is also include examples, but not talk over the example. I'll include a picture which solves the examples. Okay, so you get a general idea of how to solve a genetic variation example. So the first step is basically count the number of times an allele occurs in a gene pool. Because you see, gene pool is important as well. Okay, the second step, there's basically 2 steps. Okay, divide by the total number of alleles, okay, for that gene in the gene pool. Okay, now overall, when I talked about this, the whole idea of genetic variations, difference between how we cannot generalize every other penguin by just looking at one penguin. Like you guys can see over here, we have calculating Alex frequency or rate. Now we can take the frequency of allele as shown of frequency of allele G is equal to 7/12, frequency of allele small g is equal to 5/12, and we can find the percentage. When I talk about this, the whole idea, okay, what I'm trying to emphasize on is that genetic variation in a population increases the chances that some individual will survive. Okay, so what I'm going to do now is basically talk about genetic variation from 2 main sources. Okay, genetic variation comes from 2 sources, 2 major sources. So let's go ahead and talk about those 2 main sources. Okay, so we said the genetic variation comes from 2 main sources. The first source is mutation. The second source is recombination. Okay? Now since I already tell you the 2 main sources, what I'm now going to do is basically describe the sources. Okay, so let's start off our discussion with mutation. So what is it? What mutation can do is basically change a form of a new gene. Okay, so we don't quite understand that. So let's go ahead and put in an example into it. So let's put in reproductive cells, for example, which basically can pass on to offspring. This increases the genetic variation in a gene pool. There are many genes in each individual and many individuals in population. New mutation form frequently in gene pool. So what does that mean? To summarize mutation, well, mutation is nothing more than a random change in the DNA of a gene. Okay, so let me go ahead and erase this and put it down so you guys don't get confused. Okay, so that's mutation, and all we did was put in an example of reproductive cells. Okay, now it's recombination. Recombination. So let's go ahead and talk about what is recombination. Recombination, as you know, is nothing more than random change in the DNA of a gene. So now we come across what is a recombination. Well, new allele is a combination in the form of offspring through the process called recombination. And you guys know this because in our previous videos we talked about meiosis, mitosis, but we don't want to recall about mitosis. We only want to recall about meiosis, because in the process of meiosis, which basically, if you don't know what meiosis is, it's the type of cell division needed for sexual reproduction. Okay, so it's basically a process for sexual reproduction, and it occurs in what's called meiosis. Okay, and as you guys know, there's two parts of meiosis, which is meiosis 1 and meiosis 2. Now, if you don't know what meiosis 1 and meiosis 2 is, hold back and look at the animation of the video to get an idea of what is meiosis 1 and what is meiosis 2. Well, just to give you a hint, it's a process of splitting the sexual cells, which are gametes, like we talked about. Okay, so it's just splitting the male reproductive cell, which is sperm, and female reproductive cell, which is ova, and trying to make an offspring out of it. Okay, so let's go ahead and look at the animation and understand this lesson. And after you looked at the animation, go ahead and click the like button. It really helps me a lot. Thank you for watching the video. Let's go on to the animation part. During metaphase 1, the homologous structures are randomly lined up or assorted along the middle of the cell. The same cell, you could assort the homologous structure multiple times. The arrangement would most likely never be the same. This random assortment helps create and maintain genetic diversity. In anaphase 1, the paired homologous chromosomes are separated from each other and move towards opposite side of the cell. Sister chromatids remain attached. In telophase 1, the spindle fiber falls apart, and in some species the nuclear envelope forms again. The cell undergoes the cytokinesis, which results in two haploid cells. However, the chromosomes in these cells are still duplicated. Meiosis 2 divides sister chromatids. In prophase 2, the center chromosome and centroids move to opposite side of the cell, and spindle fiber starts assemble. If necessary, the nuclear membrane breaks down. In metaphase 2, spindle fibers align the chromosome along the cell equator, so you guys can see. In anaphase 2, the sister chromatids are pulled apart from each other and move to opposite side of the cell. In telophase 2, the nuclear membrane forms again around the chromosome, the spindle fiber breaks apart, and the cell undergoes cytokinesis. This results in haploid gametes. I hope you guys enjoyed this video. Thanks for watching.